March 12th, we'll have We Need to Conserve Biodiversity, Improving Our Teaching Will Help, and that's with Dr. Suzanne Macy, also from the American Museum of Natural History. On March 26th, we'll have Youth and AI. That's a working title from Allison Herman at Cornell University. And then on April 9th, we'll have Adrienne Lamb from Binghamton University talking about Antarctica's um, view on extreme volcanism of the past, and she is on her way back from Antarctica as we speak. And for our Earth Day event, we'll have Peter Bardaglio talking about power for the people, democracy, renewable energy, and civic action. And our final session for the spring will be Ling Wei Kong talking about using AI to study animal behavior. And our current exhibit at the Museum of the Earth is Marvelous Mollusks. And I am going to hand it off to either Warren or Rob to say a few words about Darwin Days. And I'll pull up that slide.
Thank you, Dan. I'll do that. This is Rob. So we are absolutely delighted to have Jessica Goodhart be our keynote speaker for Darwin Days this year. This is our 20th year doing Darwin Days, and Warren Ullman and I just wrote a blog for the PRI website about 20 years of Darwin Days. Many of those years have been done in collaboration with Cornell University, and every year we have a different theme. And this year, the theme is mollusks, as Don said, to go along with our temporary exhibit in the Museum of the Earth. So some of the Darwin Days events have already taken place for this year. Almost every year we have a movie of some sort at our local theater, Cinemapolis, that explores a theme of evolution. Sometimes we have panels. We always have a keynote speaker. And one of the other things that we always do is have a family day on the Saturday of Darwin Days. So this Saturday, we will have an event from 10 a.m. to 1 p.m. at the Museum of the Earth. We'll have a talk there by Rebecca Rundell from SUNY ESF, whose students are here this evening. And Rebecca will be speaking basically about the wonderful shapes that we see in mollusks and how they came to be that way. We'll also have a variety of family-friendly activities. It's open to anybody, of course. We have things that are appropriate for children, but also things for anyone who's just interested in natural history and evolution. So we hope that at least some of you who are in the Ithaca area might be able to visit that. And with that, I will pass it back to Don to pass on to our speaker.
And I will just pass it on to Jessica. Go ahead, take it away. I'm going to stop sharing so you can start. And thanks so much for joining us tonight.
Give me just a second. Give me just a second to set it up.
Here it comes. There we are.
Can everyone see that okay?
Yep, looks good.
Okay, perfect. Hi, everyone. I'm excited to be here today. Thank you to Don and Rob for the invitation. I'm looking forward to sharing some of our research with you and what I know about these crazy mollusks that steal things from their prey. And again, I'm Dr. Jessica Goodhart, an assistant curator and assistant professor at the American Museum of Natural History. Before I get into the details of these what I like to call pirates, I first want to kind of take a step back and introduce myself a little bit. So I am originally from Southern California. For those of you that are familiar with that sort of topography and the ocean there, you might recognize a sort of California tide pool-like area. So this is what I kind of grew up going to when I was a kid, going to see, all the animals, all the algae, all the different things you could find in tide pools. And it's one of the things I loved most about the ocean. being able to see all of these really amazing creatures. And this was true from a pretty early age. So I actually found recently a picture of me in a tide pool at, I think I was age 8. So I would have been something like the 3rd grade, maybe. This is me actually holding a sea slug. You may be able to tell by my face that I'm maybe not excited about that at this point in time. But it was kind of exciting to go back. We found this in my parents' garage a few years ago, and it was kind of exciting to go back and to realize that even though I didn't really appreciate what this was, I was holding this animal at a very early age where, now I study things like that. And it's kind of exciting to kind of bring things full circle. So at age 32, I replicated this image, not in Orange County, but in San Diego, California, holding the exact same species and doing my best to make the same face, although I don't have the same aversion that I used to. But in any case, when I'm thinking about these habitats where there's all these cool organisms and amazing things that they're able to do, if we think about a tide pool, you might on the surface see all the algae. There's a lot of it in this particular tide pool. But beneath the surface of the water, there's all these different organisms, including many animals, that are just trying to survive. And in tide pools, that can be a challenge for a variety of reasons, including temperature, but not just in tide pools, in a lot of other areas in the ocean, in any communities, basically what organisms are trying to do is to survive. And there's a couple of main obstacles to that. This is a drawing created by my PhD student, Lena Raubold, was basically trying to sort of show what this looks like. And those obstacles are finding food and not being food, right? So you want to be able to find food to sustain yourself, but also to avoid predators so that you don't get eaten. So in this particular image, down at the bottom, I don't know if you can see my cursor, but I'm going to do this just in case. Hopefully you can, but there's these 2 sea slugs here at the bottom. And in gray, we're showing the food that they're looking for. So this could be anemones, it could be corals, tunicates, hydrozoans, sponges. And then they're also trying to avoid the predators in this community. So these are things like fish, crabs, or sea spiders, also called pycnogonids. And so these animals are really just trying to find their way around and both avoid being prey and finding their own prey. But the cool thing about this is it's not just their own genome that's able to decide whether they're able to find prey or not. But normally it would, they use their normal chemosensation, mechanosensation to go find whatever they're trying to eat. And they use the same tools that are generated by their own genome to avoid predators. But there's actually a lot of organisms that are able to steal from their prey in order to do both of those things. And that's mostly what I'm going to focus on today. So there's usually 2 things associated with theft in animals in particular. These are basically for metabolism. So you can think of a cnidarian dinoflagellate endosymbiosis, where there's, photosynthesizing symbionts inside of, coral polyps. This is also true in things like giant clams, also in things like foraminifera. And then also for defense and or prey capture. And I'm combining those together because sometimes we're not 100% sure if it's for one or the other or both. But there are animals like this boxer crab that just attach anemones to its claws. Others that can steal chemicals for defense. Many arthropods do this. Others that steal defensive organelles, which we'll talk about later. And some that steal chemicals or, harbor symbiotic bacteria in order to produce bioluminescence. So there's a lot of different ways a lot of different things they steal and a lot of different ways they do it. You may have noticed on here, mollusks appear to be particularly good thieves. And so I want to focus on those and a particular group that I study in my lab, or that we study in my lab. But first, for those of you that are unfamiliar, maybe most of you are, mollusks are a phylum hypothesized to have originated or began in the early Cambrian. These dates change every so often, but I think right now we're in the area of 55 to 500 or 550 to 540 million years ago. And then they diversified into, as of now, roughly 86,000 accepted modern species. So these are described species. These are ones that have names and are accepted. So they haven't been synonymized with something else. And around 38,000 described fossil species, I just got this from Mollusca Base, so hopefully we're up to date there. And these include groups like chitons, worm-like mollusks or aplacophorans, squid and octopuses, limpet-like animals, tusk shells, clams, mussels, and oysters, and snails and slugs. So mollusks are a pretty diverse, large group, second only to the arthropods, which is the most diverse phylum. And as I said before, mollusks appear to be professional thieves. There's so many of them that are able to steal things, both from their prey and also from the environment around them. These include, as I mentioned, bivalves like the giant clam that are able to incorporate these symbionts into their tissues in order to benefit from the benefit from the metabolism of these animals. Also various slugs. Oops, there we go. Various snails and slugs and gastropoda. These two images don't necessarily match up, but some of them steal things like chloroplasts, nematocysts or stinging organelles, and chemicals from their prey. Not necessarily all of them do all three, but there's a really great diversity among this group. And then we have cephalopods, and this is an example from a Hawaiian bobtail squid where they actually harbor Vibrio bacteria inside of a luminous gland. And the benefit of this for them is that the luminous gland is underneath and it creates an illusion when predators look up. It sort of provides them the protection of being hidden by the sun, and effectively the light makes it seem as though they're not there. So mollusks appear to be really good at this, but within mollusks, particularly excellent models of this are sea slugs. These are some examples of in particular, different types of nudibranchs. But I want to introduce what sea slugs are and why we think that this is such a common theme within this group. So first, what are sea slugs? These are basically snails and slugs within the most speciose subclass of gastropods that are called heterobranchia. You don't have to remember that. But sea slugs are basically individuals that have a reduced or completely absent shell as adults. And I'll show you a little bit of what this looks like. But they all have larval shells as far as we're aware. So they do have shells at some point in their lives, but the adults are where they seem to lose them. So these can include things like reduced shells in sea hares and bubble snails. So for example, in the side-gilled slug, this is a group that has usually an internal shell. So you can kind of see the outline of it through the mantle here. This can also include sea hares, where they have these parapodia on the sides that close up, but the shell is inside of those parapodia. So they're kind of internal, but not 100% internal. And then on the sort of more external side, you have things like bubble snails that they do have shells on the outside, similar to a lot of gastropods, but they usually can't fully go into those shells, or the shells are extremely thin and very reduced. So for example, you can see in this shell, or you may be able to see, you can kind of see through it and see the rest of their body or their organs inside the animal through the shell. So these are what we might call sea slugs. But in addition to sea slugs that have some type of shell, we have some like nudibranchs and sacoglossans that lose their shell during metamorphosis. So they're totally soft-bodied as adults. So I want to show you this video. This is an egg mass from this species here, Bergia stephaniae. This is a nudibranch, and I'll tell you more about that later. But if you look inside some of these shells, you can see these little tiny eye spots, these little black eye spots here. And those are animals that are actually metamorphosing inside of the shell at this moment, or not at this moment, but at the moment this was recorded. And you can actually see one down here in the bottom right-ish, one that's trying to crawl out while this video is happening. And you can actually see some empty shells here too. So this is how they lose their shell as they go through metamorphosis in order to become adults that don't have a shell at all. And so because of this loss of shell, the reduced shells, there's sort of a hypothesis that essentially the loss of the shell has resulted in a diversification of the ability to defend themselves in different ways, but it's also opened up some opportunities for them to steal not only chemicals or structures for defense, but also to steal things that give them metabolic benefits as well. And I'll go through some of these and what we study in my lab so you can get a sense of the kind of questions we're asking and what we're hoping to understand from these really cool models. So first off, you guys all may have heard of these before. These are very popular. I see a lot of memes and cartoons with these guys, but that may say more about my algorithms than anything else. So these are, I think people call them sea sheep, but these are Cassasiella carotimae. You can see their little eyes here, and they have sensory structures called rhinophores here in purple. But what's really interesting are these finger-like projections off of their backs. Those are called serrata. And inside each of these serrata is a branching of their digestive system. And that's what the green is that you see inside. And that branched digestive system has cells that are able to pick up these organelles that are important for photosynthesis, chloroplasts, from their algal prey. And that's why essentially these structures are green, because those cells are harboring those chloroplasts for these animals to use for photosynthesis. Not all sacoglossans do this. That's the group that this animal is in, but many of them do, and they often have sort of differing levels of their ability to retain these for metabolism. The research on this is still fairly fresh. There's been a lot of people working on these systems for a long time, but because of new techniques like single-cell RNA sequencing and other types of physiology type techniques, we're able to start learning new things about how this actually works in these slugs. So for example, Dr. Corey Ellard at Harvard University recently, or last year, published a paper starting to think about how these slugs are able to steal chloroplasts. And a couple of the things he found were that they have these special organelles inside that are essentially phagosomes or sacs or pockets where the animals are able to store stolen chloroplasts inside of the cells. And they're able to maintain an environment that allows these chloroplasts to continue functioning, at least for a time. But what's interesting, at least in this animal that they were studying, which is called Elysia crispata, they actually found that instead of what you might expect, where when they starve the animal, the chloroplasts actually become more important and provide metabolism. In this particular animal, the stress of starvation induces chloroplast digestion. So for whatever reason, in this particular taxon, the chloroplast doesn't provide enough nutrition, or it's not seen as enough nutrition by the animal for them to try and keep them going when they're starving. So instead, they just digest them. But what's really interesting is he started to find these similarities to what we see in cnidarian dinoflagellate endosymbiosis, which is much better studied, partly because of coral bleaching and the fact that understanding the symbiosis in that system can have big impacts on what the coral reefs look like in many areas. So in my lab, I have a newish PhD student that's interested in the mechanisms of targeting, internalization, and storage of these structures. So on the left, these are Elysia chlorotica that Dylan down in the bottom right here has collected, both from, I think these ones are actually from Florida, but you can see they're very green, and that's because they're full of chloroplasts. And if you look, these animals can actually open up. And this opening of the parapodia is basically how they get the most sunlight on the areas where the chloroplasts are to get the most out of their photosynthetic capabilities. But one of the things that we're interested in is actually on the sort of earlier side, which is in these first two pieces of chloroplast recognition and phagocytosis, or internalization of those chloroplasts. Because the cells somehow have to be able to identify these organelles as important or valuable in order to target them for taking them into the cell. And once they have to have some mechanism, most likely some sort of conserved mechanism for internalizing that structure and keeping it functioning for the long term. So my PhD student is interested in understanding how are these chloroplasts recognized by host cells, what pathways are used for internalization, and then ultimately, these will take a long time to answer, but what genes are important for storage and maintenance of these chloroplasts. Okay, so in addition to our understanding of of this, what we call kleptoplasty, or the theft of chloroplast uh, for metabolic benefit. That's actually quite rare within sea slugs. The vast majority of slugs are actually able to steal mostly for defense. So again, another drawing by my PhD student, Lena. She had a couple of nice ones, so I borrowed them. And this is really just showing that these slugs don't necessarily all steal everything, but that there are certain taxa, like this species here feeds on sponges. This species here is going for something like the stinging organelles and hydrozoans or anemones, basically to highlight that they have different preferences and they often steal different things. So they seem to be specific to some extent in terms of their ability to steal. And often that results in some level of prey specificity as well, though Lena is also looking into whether that is true. Okay, so some sea slugs do steal defensive chemicals. So they're stealing, in this particular case, this is what we call a Phylodesmium limbahense. The animal is actually right here. This is the slug. And you might notice that this thing around it, which is actually the soft coral, looks a lot like the slug itself. And that's because these particular animals seem to be very cryptic on their prey, which is usually soft corals. But inside of these projections, these are again cerata. We'd call them the same thing as any other group. Inside of these cerata, we actually find that they have these special sacs that are where they store chemicals. It may look like they're empty in this histological section, but they're not. They actually are full of chemicals that they're stealing from their prey to ostensibly use for defense. You can see another good example of this crypsis in this bottom right image, where you can see the animal is actually up top here and the soft coral is around it. In addition to animals like the Phylodesmium, there's also other types of slugs called dorids. These are, the vast majority of these appear to steal chemicals, though not necessarily all of them. But what you might note is that it actually looks quite different. So these animals don't have those cerata, those projections off their backs. So instead, they eat things like sponges, like this Phorbus fictitious here, and they steal chemicals from that. And they actually place them in usually sacs around the margins of their mantle or around the edges of their body, basically, as their means of defense. So it's very different from the last slide I was showing you, where the animals kind of have it in these special structures. Instead, they have them in special glands, but they're just sort of around the edges of the animal. And in this particular case, it's a way to highlight the chemical from the animal is actually on the right-hand side here, but in the slug, it's actually a little bit different. So the structure of the chemical is to some extent modified in the slug sometimes, but not all the time. Okay, and so the last type of theft that I want to talk about is, to me, is the most exciting because it's what I've studied for most of my career, which is these slugs that steal stinging structures from their cnidarian prey. So cnidarians are things like jellyfish, Portuguese man-o-war, anemones, things like that. And if you've ever been stung by a jellyfish, you know how painful it can be, like this example on this person's leg here. It's not my leg. I've been asked that question before, so I just want to get out ahead of that. Thankfully, I have not been stung by a jellyfish, knock on wood. But the way that this works is they have these specialized organelles inside individual cells, and these fire. Like this on the left, you can actually see the capsules and these harpoons, which we call the tubules, these fire out of the cells in order to inject a mix of venoms into either their prey or predator. This is a very slowed down video. It's normally much, much faster. You think like bullet speed. But if you look closely, you can actually see what looks like a liquid coming out of the tips of these. And that liquid is the mix of venoms that I was talking about. So it's cool to be able to kind of see what that looks like. So cnidarians are largely very well defended if you think about this kind of unique ability that they have to create these special structures. But slugs somehow not only are able to eat them despite this, but then they actually steal those weapons from their prey. So like this animal here, this is a undescribed species of Abronica. The head of the animal is over here. The tail is on the right. And again, these special structures called serrata seem to be really important for this type of theft because inside of each of these serrata is again that branching of the digestive system, similar to what we see in cecoglossans. And these singing organelles or singing structures are stolen and stored at the very tips of these. So the digestive system branches, and in the very tips here are basically these specialized organs called cnidosacs. And these are where the organelles are stolen and stored. And if we want to see what this looks like, on the right is an animal, um, a juvenile animal where we can see the actual contraction and expansion of the digestive system. So if you see, we have it's kind of a mix, but we'll see that's expansion to contraction to expansion again. And that's effectively how everything moves through their digestive system. It moves super fast, at least early on in digestion. And if you look closely, you can see at the very tips of each of these serrata, these little sacs that seem to have what look like oval type things. And those are those singing structures that they've stolen and stored inside that cnidosac. And we know in these animals that it can be used for defense and prey capture, although not all of them use it for prey capture. It seems to be kind of specific to certain groups. So for example, in Bergia stephaniae, we can watch as if you harass the animal, they sort of orient their serrata toward that stimulus. And what we sort of hypothesize is happening is that's directing the nematocyst towards the place where theoretically the predator might be. And ideally for the animal, the predator might take a little bite and get a mouthful of nematocysts. And hopefully that will be enough for the animal to get away. And the real benefit of how these organisms sort of perform these functions is that these serrata can actually be detached and they can regenerate them. So it's a really cool system where they're able to just very easily regenerate these defenses. Not necessarily super fast, still takes maybe a month, but they still survive and are able to continue defending themselves later on. And then on the flip side, we have prey capture. So in this Glaucus atlanticus, this is, I think, like a blue sea dragon is what people call it. The serrata are actually on the sides. And this is a little piece of fish being dropped on the animal to show you how the animal is going to orient the serrata towards what appears to be prey and sort of holds onto it. The nematocysts are theoretically fired in order to stop the whatever it is from moving, and the animal is able to eat from there. So we have these really cool insights into how these are potentially being used inside different species. Excuse me. So in our lab, we study this theft in a species called Bergia stephaniae. That's this animal here on the left. It's a native species to the Florida Keys. And it's actually a really fun one to find in the wild. They're super easy and chill. I really enjoy it. But I really like the species for a number of reasons, not least of which because my master's advisor actually named the species after his wife, Stephanie. So it's kind of nice that I get to work on it. But in addition, they're able to they're able to eat a species that is considered actually a pest in the aquarium trade. So on the right here is these Bergia animals sort of surrounding what was an anemone. And that anemone basically gets destroyed over the course of a couple of hours. This is a sped up video in this case. And so they're actually quite easy to keep in the lab because these Exaptasia anemones are pests in the aquarium world. We can get them very easily and grow them very easily in the lab. And it allows us to keep the slugs as well. And because we have them in the lab, we can watch how these structures move in the digestive system, similar to what I showed you before. But this is just a single cerus, and the digestive system is down here, which if you look closely, you can actually see these oval structures. These are the stinging organelles. You can see them move up into the cnidosac up here and back out again. And for us, this was really intriguing because it suggested that there's an ease of movement between the digestive system and the cnidosac that basically makes it so anything could potentially get in there. And we do see that. So if we look at the dinoflagellates, these are dinoflagellates that are usually inside the anemone that the animals just end up eating by proxy. They can also move up into the cnidosac. And because those symbionts can move into the cnidosac as well, what that suggests to us is that the cells that are picking up these stinging organelles they're actually interacting with a lot of stuff that's in the digestive system, including symbionts and other types of structures that the anemone had in its body. But somehow these cells are actually able to target the stinging structures, very similar to what we see inside in kleptoplasty or the chloroplast theft in Sacoglossans. So it's a really cool similarity that they share, despite the sort of morphological differences. And as I mentioned, we're interested in understanding sort of the genetics of these types of things as well. So we found that at least 2 immunity genes are expressed in the cnidosac. This is work done in part by Rebecca Lopez Anito, my PhD student. And I'm going to show you how we know that they're expressed inside the cnidosac. So on the left here is an HCR image. HCR is a form of in situ hybridization. Basically, it's a way for us to see where genes are being expressed or being used inside of the animals. So on the outline here in gray is showing you the outside of the tip of a cerus. And you can see the digestive system it's like a slightly lighter gray in here. And where all the color is, the outline of that is the cnidosac. So those cells where all the color is are where those genes are being expressed and where we're seeing where the nematocysts or stinging organelles are being picked up. And just to kind of tell you what these genes are, one is a C-type leptin macrophage mannose receptor, basically a receptor that we think, or in something like humans, would be used to maybe target bacteria for digestion or destruction. And it's extremely highly expressed in these tissues, and we think it might have to do with their means of targeting nematocysts or stinging structures in particular, because it doesn't seem to be expressed, at least not in high concentrations in other tissues. And then we also see this collagen 8 alpha 1 gene expressed in exactly the same cells in generally the same places, though with apparently higher expression, where you can see where we think that this gene is actually used to help with the remodeling of the cells, because these cells have to expand to accommodate these stinging structures, especially for some that are quite large. So we do a lot of lab work with this species in the lab, but one thing I really enjoy about my job at the American Museum of Natural History is that I also get to do a lot of field work. So we also are able to collect animals from the field to study, because if you just focus on one species, you're not able to really get the full picture of how different organisms are doing the same thing and how that function might have evolved from animals that don't do that. So we do a lot of field work, including scuba diving in different places. We go snorkeling and dock collecting, which is basically looking on the sides of floating docks for these animals. Also tide pooling, which I showed you some tide pool images. And then we process these animals by taking live photos, because their color gets leached out when we fix them. And by sometimes collecting data on what they're potentially eating, by collecting data on or videos showing them eating something, or collecting fecal samples, which is something my PhD student Lena has been doing a lot of to try and better understand the diets of these animals. So we're doing a lot when we're collecting them from the field to study, but one of the things we're really interested in is sorry, I went too far forward. We're able to collect them basically in oceans everywhere, which is a really nice advantage. You might notice a higher proportion of tropical locations in my experience with fieldwork. That's not just because it's more comfortable. The species diversity does tend to be higher in those areas, but in general, you can find sea slugs anywhere in the world as long as it's in the ocean. And what we're really interested in when we're going to collect all of these species is not just kind of collecting anything and everything that we can get, but in using these to try and better understand how these animals are related to one another, so that we can reconstruct how many of these features, like different types of theft, evolved in sea slugs. So this is just an example of a phylogenetic tree that I generated in my PhD. I won't go into too many details. You don't need to worry about that here, but it's just to show kind of what we're using these data for, or these animals for. And in this particular case, we were able to show that the theft of stinging structures actually evolved at least twice within nudibranchs, and particularly within this group called Cladobranchia, which is a group of nudibranchs. And that includes Aeolidida, which is where Bergia stephaniae falls, as well as the Abronici I showed earlier. And then also Hancockgia, which is a much smaller single genus of animal of nudibranchs, where this ability appears to have evolved secondarily, or a second time, sorry, rather than just this initial one on Aeolidida. And one of the things we're interested in is not just, oh, it evolved a second time, but were they using similar functions? Was there some sort of background within the sea slug group that made it more likely for something like that to evolve? Or are they doing this in an entirely different way, which would also be really interesting. But one of the challenges we have with Hancockgia is it's actually quite poorly described. So it's important as a convergent origin of nematocysts or stinging organelle sequestration, but there's currently only 6 described and named species, which, that doesn't necessarily mean that's a bad thing, but there's already an estimated 7 to 10 known undescribed species. So already my PhD student, Lena Raubold, again, she's going to be working on basically trying to describe those additional species. And we hope to more than double the number of known species in this group with this particular work. And so it's really important that we think of not just the sort of detailed mechanism, molecular lab stuff that, we do sometimes, but also to think, like, step back and look at the broader picture in order to really understand how these things evolved. And the really cool thing about Hancockgia is it does appear to be different in terms of, at least in terms of morphology, because unlike things like Bergia, they have one cnidosac in a single cerus, but Hancockgia, they can have multiple. And in this animal, it's showing that there are 3. So we already know that there are some differences between these 2 groups. The other thing I wanted to plug is that especially for nudibranchs and sea slugs, many citizen scientists or community scientists assist in these efforts. So when I'm going to new places to collect animals, often I'm connecting with local community members through things like iNaturalist, through websites that are specific to certain areas, to find people that know where the animals are, to find people that are excited to learn more about them, and to help them to identify what they have in their area as well. So it's a nice mutually beneficial relationship when we work with folks that sort of create these types of websites or contribute to websites. And luckily, sea slugs are largely very charismatic, so scuba divers tend to really love them, which is good for us. I just want to plug, if you aren't already, to please consider helping describe biodiversity, even in your backyard. All you have to do is download the iNat app, record your observations, and sort of contribute to the community. And feel free to, if you ever see any sea slugs, add them as well. So I just wanted to kind of end this part of the talk by highlighting that in my lab, we're really interested in this idea generally of how this biological theft evolved. And again, we're looking at it in these multiple systems, this ability to steal the chloroplasts from algal prey, stealing these defensive structures from cnidarian prey. But in order to do that, we have to be able to look at multiple levels of organization, from the genes, proteins, and cell types, all the way up to different tissues, behaviors that they use, and ecology, in order to fully understand the picture in which these types of abilities evolved. Because in order to reconstruct them, you have to understand how all of these pieces became integrated over evolutionary time, which is a long time. So there were a lot of different things that happened at those given times in history. But I want to end the talk on this question. If you're listening right now, you're probably a person that already kind of appreciates to some extent why understanding basic biology like this really matters. But even if you're not, that's totally okay, because I think It's kind of our job as scientists to try and make that connection clearer. So I wanted to highlight a few things that I think are relevant for my particular systems, and then thinking about basic biology generally in terms of why it matters. So first off, very directly, there have been chemicals used from sea slugs, from nudibranchs for drug discovery. So an example of this is a drug called Zolipsis, which was generated based on chemicals from multiple marine invertebrates, including a nudibranch. That's been tested in multiple clinical trials, or in clinical trials to treat multiple myeloma. Some of these don't necessarily make it all the way through, but basically it's just to highlight that marine invertebrates like sea slugs are actually a really great place to look for these types of things. In terms of the actual stinging structure, oh, sorry. Okay, that's when I had a question. In terms of the actual stinging structures, people have been actually looking into whether these would be useful for drug delivery. You could imagine, for example, in a capsule where it has the harpoon, normally there'd be venom inside, but if you were to add some sort of drug, which here is in these little green like ovals here, maybe you can create this drug delivery system where the harpoons actually inject the drug into a particular area of the body. So in this example, people are trying to use it for the skin to inject certain drugs deeper into the epithelium rather than just on the surface with a topical cream or something. But when I think about this, I think about how can the slugs be useful? And I can imagine sea slugs helping with stability. So for example, these structures have to get through their entire digestive system without firing. How does this happen? And is there a way we can learn more about either the stinging structures or about how the slugs are able to prevent those from firing by studying these systems? And then beyond that, if you're able to create that stability, you might be able to not just be able to deliver drugs to the skin, but be able to deliver drugs to the different parts of the body, especially if you know how are the slugs targeting these structures. Can you use receptors from certain organs to allow them to target these stinging structures as well? Definitely a way down the line, but it's things like that that I think understanding basic biology it's really important for kind of coming up with creative solutions and innovative ideas. And then I want to end on just sort of this very basic idea that we are animals and we are part of nature. And I think us understanding nature in all its forms is really fundamental to our survival so that we can both protect it, but also appreciate how we are a part of it as well. And, there's always, if that's not enough for you, we can always use it for ourselves, I guess. But I hope that this is enough. In any case, I just want to end there, and I want to say thank you to all the folks that have helped with this work, and all the people in my lab or that have been in my lab. I've got a great lab. We're doing some cool work, and I'm excited. I'm hoping some of it comes out this year. And I'm really grateful to all of them for all of their hard work. And I'd like to thank you all for your time, and I'm excited to answer questions.
Thank you, Jessica. That was great. And there's already a question in the chat, which is, how does the animal deal with the remnant discharge cell after the discharge?
So I guess I'll sort of take that in two parts because I'm not sure exactly what point they mean. But early on in their digestive system, some of these stinging structures can fire, and they have these protected cuticle linings early on in their digestive system that protect them from these stinging structures firing. And so what ends up happening is those pieces can get digested, or they can just be released in their feces. So it's like sometimes they just go completely through the digestive system without being digested or destroyed at all. If you're thinking about on the other side of things, where after they've taken these stinging structures, then what we think is happening is actually that they use the nitosac as a muscular sac, and that they're able to kind of squeeze those out. We see this sometimes when we do some preps where they can kind of squeeze out these stinging structures altogether, so they're not actually inside the animal at that point. They're just on the outside. Hopefully that answers your question.
That's good. And Gustavo has a hand up and after Gustavo, there's another question or two in the chat. So go ahead and unmute yourself, Gustavo.
Yes. Doctor, excuse me. Do you say about the different gasterops, kind of gasterops get immunity, but I don't know if it match with symbiosis.
Sorry, can you repeat your question? Which ones?
No, sorry. The gasterops get immunity by symbiosis?
No, they don't really get the immunity. The immunity genes I was talking about are genes that they produce. Does that make sense?
Well,
Those are their own
well,
genes from their own genome.
well, okay, doctor. No more questions.
Okay. Next up in the chat is, are there freshwater analogs to sea slugs? From Jim.
Freshwater analogs. No, well, I mean, there are terrestrial slugs. That's maybe the closest, but otherwise not that I know of. Certainly not in terms of gastropods. As far as I know, most of them are shelled.
And Paul asks, are there opportunities to work in your lab?
Perhaps this coming year, yeah. I wasn't taking PhD students this year, but certainly next year. And if you're ever interested, just reach out. We can talk about potential opportunities, and I have some information on my lab website also.
Awesome. And Jan asks, I don't know how to pronounce that. Can data sacks contain undischarged nematocysts cysts or fired nematocysts?
Nematocysts.
Thank you.
Yeah, no worries. They contain undischarged nematocysts, so they're ones that they can still fire basically because they only fire once. So once they've been fired, then they're basically no longer useful. Yeah.
And Gustavo asks if he can keep in touch. And there is Adria asks, when you say you fix the animals you acquire, are you euthanizing them for the samples?
Yeah, it's a bit of a misnomer to say fix. Yes, we are. We're euthanizing them and we preserve them, and usually ethanol, or what we fix in formalin, which means we're just sort of initially preserving them, but then they get transferred into ethanol. And for the most part, that's what they stay stored in for all of these animals. That allows us to keep studying them in the future.
Okay.
But we do have live animals in the lab, as I mentioned, so not all of them are dead.
Yeah. And Haley rather asks, is there any way for this behavior to be represented in the fossil evidence? If so, what form?
I don't know of any sea slug fossils. I think the only thing that's been found is really like radulae, which are they have these sort of scraping teeth structures. That those are called radula, or it's called radulae that they sort of use to scrape bits of tissue off that they eat. But I don't know that you would be able to identify those. Certainly, you wouldn't be able to identify those as like coming from specifically something that steals. But I don't know that you'd be able to identify them specifically as sea slugs either. And they're all very soft bodied, so it'd be pretty hard to tell, unfortunately. Yeah. My lot in life is to study the only mollusks that we probably can't get that information from.
Yeah. And Lorraine asks or shares an anecdote with too many words that I can't pronounce for me to
I'll
read
leave that.
aloud. So
Okay, I'm looking at it.
Mhm.
Yeah, that's a great example. Yeah, it was the Serrata, I'm sure. But that goes to show that the predators are, they can still eat them. It's still possible, certainly.
Okay, other questions? Rob's got a hand up. Go ahead, Rob.
Only my doctor. Can I keep in touch with you by
yeah.
email?
Sure. Yep. My emails, you can find it on my website.
Can you share me, please?
I can put it in the chat too, yes.
Thanks.
Mhm. Go ahead, Rob.
All right. So I thought it was fascinating that some of the research that you were doing is based on undescribed species, which I think says something about the the number of undescribed species of sea slugs that I imagine that there are. And I wondered if you could, you talked about it within one genus, I think, but I wondered if you could talk a little bit more about how well sea slugs are known taxonomically as a group, and how and to what degree there are new species being found on a regular basis.
Yeah, I mean, so there's something on the order of around 3,000 named, I think, accepted species. Um, that's definitely on the low end. So I can really only, beyond that, speak from experience. I don't have the numbers in terms of how many are being described regularly. There's certainly a lot of people that are regularly describing new species of slugs. But I think the problem is we're finding them faster than we're describing them. So, for example, I went to Perth, Australia in 2023, and I estimated maybe a third of those species are potentially new, despite the fact that there have been people working in Western Australia um, fairly regularly. And this is something that a lot of my colleagues are finding, especially in the Indo-Pacific, where no matter when they go, no matter how many times they've been to a location, they're always finding something new. We're not really reaching those points where we're like, oh yeah, we've gotten all of the diversity from this place. So I think like the Philippines and Indonesia are good examples of this. Um, but I mean, based off of how many are being found all the time, I would estimate somewhere between probably 7 and 10,000 species are probably likely in existence. I think we also tend to focus on these big, brightly colored ones, but there's a lot of maybe less exciting ones that are very, very tiny that are certainly mostly undescribed in my
Yeah.
experience. Yeah.
So that's super interesting. I do ask in part because there are so many students uh, on this call who I think may be surprised to find how much of the natural world is unknown even at a basic level. It may seem like we know it so well, but for groups like this, there's a lot of work to do.
Yeah, definitely.
And there are a couple more questions in the chat. Are all kleptoplastic nudies colorful or are some drab?
That's a good question. Some are definite. It depends on what you mean by drab. I would say a lot of times they're still very pretty, but not all of them have those really bright colors. Certainly the really, really tiny ones usually are not. They tend to be like brown or green, but I think they also don't need the bright colors. So often the bright colors are considered sort of a sign of what we call aposematism, where they're basically a sign of whether they're dangerous or not. But if you're small enough, that may not be as much of an issue. Maybe you're more interested in hiding. So it's definitely a range. You can even see on this slide that some of them are not as brightly colored. So like, I don't know if you can see my cursor anymore, but uh, this animal here is probably a sequestering slug, and it's pretty brown here as well. But there are some more brightly colored, more interesting ones, like this purple and orange one, for example. So
Even the drab ones are
definitely a
pretty.
mix. Sorry.
Even the drab ones are pretty.
Even the drab ones are pretty. I think so, but I'm maybe biased.
Nah. So that question was from Timothy. Erica asks, do any species have the ability to utilize both chloroplasts and stinging cells?
No, but there are some that steal stinging cells and also steal, so they eat things that have dinoflagellate symbionts, and they sometimes will steal those. So they're like third degree pirates because they're stealing something that's already been stolen. Um, so in those cases, some of them can do both, but it's not super common that they do more than one.
And Jennifer says, thanks for devoting time to why learning basic biology is important. And Mina asks, see many of them, or this isn't a question, I guess. I see many of them are red in color. So are they chemically defended? It is a question. Sorry. Yes.
In some cases, yeah, but to be perfectly honest, like for a lot of these species, I would say that probably hasn't technically been studied. So I think for a lot of the species that are brightly colored, we sort of make assumptions to some extent about whether they are chemically defended or not. Um, but certainly for a lot of brightly colored ones, particularly things that are like black or red, you do see a lot of clear signs of chemical defenses. But it depends. If it's a red aeolid, then that's probably more likely to have the stinging structures.
And then Sebastian asks, when did sea slugs first start stealing from their prey? Of course, soft-bodied animals don't leave behind fossils, but maybe we have a clue from taking the genomes of multiple species and comparing them with each other.
Yeah, so I mean, based on phylogenetic evidence we have now, I would say, I mean, I would mostly be guessing because we don't have good evidence in terms of like when those sorts of things actually happen, like the timing of divergence or timing of diversification or timing of origin. But probably sometime in the last 200 million years is likely when that happened. There have been some estimates that nudibranchs as a whole, so that's the whole group, evolved around maybe 250 million years ago. So somewhere after that time, but probably in the past 200, 150 million years is when at least one of the origins was. Certainly in Hancocky, that second origin, it was probably much more recent, but we don't have any sense of when that might have happened. No fossils, so makes it harder.
Yeah. And Eileen asks, what is their size range?
That can be a huge, oh, it really depends. So we collected in American Samoa, probably one of the smallest ones I've ever seen, and my PhD student Rebecca actually found it, and it was smaller than a millimeter. Just a tiny little orange guy. But there are some species that can get up to like near a not a full meter, but like
Oh, wow.
at least like a foot and a half, like half of a meter, something like that. But that's very uncommon, I would say. That's really big. I have a good picture of my foot next to one, and it's like a pretty good size, about the size of my head.
Cool.
So they can be quite big, but that's not
So,
that common. Most of them are very small.
yeah, that sounds a little creepy, but but also cool. Any other questions? Pretty good collection. Well, if not, we'll say
I have another question.
sure. Go
In this
ahead.
case, I see that gasterops or different kind of gasterops are similar to gasterops in Mexican Gulf or in
Mhm,
the Pacific Sea, in special near to the coast of
mhm.
Oaxaca or Acapulco. In this case, in this coast, I believe that are similar to the gasterops of Central America or Caribbean. In this case.
There's some that are similar. So a lot of times they look sort of externally very similar. They don't necessarily, but they're not always the same species. So usually, I would say in Baja, you might find some that are very similar to what you might find in California. There's some that go down that far, but a lot of them tend to be a bit more tropical. So you'd find some that are more similar to Central America. But there's definitely no overlap between the species on the Pacific coast and in the Caribbean, except where there's been sort of introductions. But they often look very, very similar, which makes it difficult to differentiate. So there have been cases where a species has been described as cosmopolitan, meaning it's found, say, everywhere in the tropics. And then we do the genetic work on it, and we find that it's actually like 6 or 7 different species that we thought were all the same because they just looked very similar. And these are scientists making these decisions. So like it definitely happens that there's some that look super similar despite actually being genetically very different.
And Eileen asks, are they found throughout the ocean or just near shore?
They're definitely found throughout the ocean. The greatest diversity tends to be much more shallow. So, say within 20 meters or 60 feet, you might find the most diversity. And the diversity is higher in the tropics generally. But they are found throughout the ocean. So there are species in the deep sea that feed on what we call octocorals. There's species in the Arctic, in the Antarctic. There are also some that swim in the open ocean. They look kind of like fish. I can put the name in the chat if people are interested because those are super cool if you look at them. They just sort of are flattened slugs. So if like this is their face, they just get flattened this way. And then they can kind of move very similar to how fish move, which is pretty cool. And they bioluminesce also.
Any other questions? Well, thank you for a very interesting talk, and we'll see folks again in a couple of weeks for talking about brain science in the real world with Ron Bonstetter and Dave Gosling.
Thanks, everyone.
Thanks all. Have a good night, everybody. Thank you so much, Jessica.
Yeah. Do you need me to stick around, Don, or?
Nope. If you'd like, I can send you an attendance list. We did break the record tonight. We were the biggest number I saw was 125, which is at least 20 more, I think, than the biggest crowd we've had.
Cool. Yeah, it's not necessary, but thank you. I appreciate it. And thanks for the invite. I appreciate that. If anyone has questions, feel free to share my email.
All righty. Thanks so much.
Cool.
Thank you, Jessica.
Bye.